Rosenblad, who's in the active lab at UC Berkeley. Tell us about adaptation and growth.
Thank you. Excited to be here. And today I'll be talking about the issue of adaptation to different facets of climatic stress across a spatial gradient. So specifically trying to deal with both drought and freezing, and asking whether alleles that seem to be, or versions of a gene that seem to be good for dealing with one, seem to be good or bad for dealing with another. I want to start out by acknowledging the undergraduates who did really awesome work on this, as well as our funders who are supporting the work. So I now want to build from the premise that success in assisted gene flow is often going to be predicated on our ability to maintain resilience to multiple forms of climatic stress. And the two that I'm concerned with right now are summer drought, which we've been hearing a lot about, and I imagine is probably familiar to many folks, and spring freezing events, which we probably hear less about. We saw some really neat examples of that in Joe Anderson's talk yesterday. Just as a sort of refresher / introduction to that issue, the basic idea is that here in the temperate zone, a lot of plants over the winter will go basically dormant or become acclimated to cold, which means they can deal with pretty extreme cold temperatures as long as it's predictable. But as springtime temperature regimes get more erratic, the plants can get tricked by an unseasonable early warm spell that tells them, okay, time to start growing and dropping your defenses. But then when it gets cold again, they get hit with a damaging freeze event. So both of these types of events occur now across much of our western landscapes, and in some places they're expected to intensify with climate change. So if we're trying to think about assisted gene flow strategies, we want to help populations maintain resilience to the full suite of stressors they're going to be experiencing both now and in the future. And I would argue that in most cases, that's usually going to be a pretty multifaceted set of climatic stressors or shock events. So to me, that begs the question of whether we might be able to find genotypes somewhere within our landscape of interest for sampling source material that are going to be resistant to the full suite of stressors that we care about. Or on the other hand, where there might be some unavoidable trade-offs, which we would certainly want to know about as we build our sourcing strategies. So just to lay out the two sort of alternative hypotheses in that regard, on one hand, you could have a trade-off whereby different forms of stress favor sort of separate, mutually exclusive genotypes and phenotypes. And this is sort of a common paradigm in evolutionary biology where we think that sometimes, you've got one limited metabolic budget, you invest in one thing, you can't invest in another. Alternatively, we could have a synergy whereby some anatomical structure or physiological process actually does a good job of dealing with two problems that we might think of as separate. So thus far, this issue of trade-offs has been studied primarily from a very kind of targeted, trait-based, often common garden study framework where we're learning a lot about a relatively small number of traits that we think are probably important, and in many cases are. But what I think is sort of understudied is maybe a complementary approach where we step back from thinking about particular phenotypic traits and just ask about whether across the genome, do we see that alleles or just sort of versions of a gene that are favored by one form of stress are the same or different from what's favored by other forms of stress. So that's kind of coming back to that trade-offs versus synergies question. And just to kind of outline, this is just a cartoon, these aren't real data, but just to outline what a study like this might look like. At the top here, I'm sorry, my slides are formatted differently now, but that's just a trade-off. And so here's what data for one example SNP, or just basically a point in the genome where we've got variation, might look like. So as you look across a spatial gradient of stress that are more stressful with respect to drought, you might see that the frequency of some allele goes up. But then as you look across a freezing stress gradient for that same allele, you might see that goes down. And then, all the way at the right there, if you're looking across all the points in your genomic data set where you've got variation to work with, you could look at the effect sizes for little graphs like these and see what the association is. And what we're seeing here is that alleles that tend to increase with drought stress across the landscape tend to decrease with freezing stress. So sort of think again, coming back to that idea that if you're good at one thing, you're not going to be so good at the other. The alternative here, the synergy scenario, would be both stress gradients showing the same sort of direction of response of allele frequencies. And when you look across all the SNPs in your data set, you would expect to see a positive association, whereby alleles that are good for one are good for the other, bad for one is bad for the other. So I'm going to step away now a little bit and just talk about the study system in which I'm investigating these problems. It's Lemon's willow, or Salix lemonii, up in the Sierra Nevada. The habitat that I'm interested in is these montane meadows that are just very sort of culturally important, important for lots of reasons. This particular species is used a lot in indigenous cultural practices, and it's really good for meadow restoration projects, because you can just kind of haphazardly hack off willow stakes and stick them in the mud and get a new willow thicket next season. From a climate change perspective, we're probably most concerned about this species from the perspective of intensifying summer drought that we've, in this region we've all been hearing about that, and so I won't belabor that point. But just from sort of my own natural history observations and a few other folks that I've chatted with, I think we also need to be paying more attention to this issue of increasingly erratic springtime temperature regimes that can really sort of trick the plants into growing too early and getting damaged. So if we're thinking about management strategies and we're willing to consider assisted gene flow, I think it comes back to that question of whether we might be able to find, predictably on the landscape, a place to go and get these sort of multi-tolerant or polytolerant genotypes. And the existence of populations like that is going to be predicated on the extent of trade-offs versus synergies from that little cartoon framework I laid out before. Just a quick orientation to the geography of the system here. I'm, at least for now, focused on Tahoe National Forest, which is this part of California right here. Got a landscape genomic data set that I'll show you in a minute. It comes from these 40 sites here spread across the national forest. And those same 40 sites are plotted in climate space on the left there with respect to two key climate variables. On the x-axis, that is, at least for now, my index of drought stress. It's basically April snowpack, which the idea there is if you've got lower snowpack, so left side of the graph, there's less meltwater, which means less soil moisture and things like that during the growing season. A nice thing about this system is that that variable is also very tightly correlated with other facets of drought we might care about, like vapor pressure deficit, maximum temperatures, all that stuff. It's kind of a catch-all drought axis. The y-axis is my index of exposure to spring freezing events. Right now I'm just using minimum May temperature. I would like to revisit this with something more sophisticated, like the growing freezing degree days after snowmelt that we saw earlier, but at least for now this is, not to tip my hand, but showing some patterns that are at least consistent with the adaptive story we might expect. So just to lay out the approach in brief, I didn't know this was animated, but I sampled 184 individuals across those same 40 sites that I was showing you a moment ago to generate, at least as a first pass at all this, fairly modest dd-rad-seq genomic data set. The idea there is this what they call like reduced representation, not trying to sequence the whole genome, which yields about 5,000 to 10,000 points of variation in the genome, depending on how you do your bioinformatics. I'm characterizing the climate of each of those sites with respect to those same two key variables I mentioned a moment ago for the indices of drought stress and freezing stress, as well as a bunch of other stuff that we want to control for kind of in the background of the statistical models. Happy to answer questions on that. I just want to flag that David and I had a paper come out last year with some common garden data showing that both of these spatial gradients show ecotypic variation in physiological traits that sort of make adaptive sense. So that's our reasoning behind highlighting these particular two variables here. So the statistical approach is kind of stitching a lot of different parts together into one big analysis where in part what I'm doing is for each SNP, each individual point in the genome where there's variation, I'm modeling the effects of those two key climate variables on allele frequencies. And then at the level of the whole data set, all of the different SNPs, I'm also modeling the association between those to see whether we have a positive association, which would be consistent with that synergy model I was talking about earlier, or whether they're negatively associated, suggesting that the trade-offs might predominate. So here's the data. These are very preliminary, and this is just kind of modeled after that structure of the cartoon I showed earlier. So this is just one example SNP that I think is fairly representative in that the effects of most the effects on most SNPs of these variables tend to be modest, but they also tend to be in the same direction. So this is an allele that happens to increase along both our gradient of drought stress and our gradient of freezing stress. And then when you zoom out and look across the whole data set, which is what we're doing on the right there, I'm not using points anymore because there's too many of them, so the color just shows you the density of how many points you would see if I plotted them all. And each point is just one SNP from the data set. The X and Y axes are the effect sizes from graphs like these. And that red line there in the middle is showing that there's a positive association, such that alleles that tend to increase with more drought stress also tend to increase with more freezing stress, and vice versa for decreases. So there's an awful lot of scatter around that relationship, but we have a pretty clear indication from the super skinny uncertainty bands around that line that this is a synergy rather than a trade-off scenario, such that alleles which are good for one do tend to be good for the other on average. So just to wrap up, we've got some evidence that adaptation to drought and freezing across the landscape is synergistic for this species, which means that these two different forms of stress tend to favor, on average, the same alleles. What that means from a management perspective is that sort of dually stressful sites, in other words, ones that are in this part of the climate space, stressful drought regime, stressful freezing regime, those are in the yellower colors. Here they are on the map. These might be good places to go if you're looking for genotypes that are going to be good at dealing with both forms of stress, maybe to use as part of your sourcing scheme for an assisted gene flow project. I'm super curious about the physiological mechanisms behind this synergy to the extent that it's real, and I think, as a first guess, I might think that it's something to do with having more osmolytes or just more molecules floating around in your plant cells, which, based on work from other systems, we think might be good for dealing with both drought and freezing in some situations. So I will leave it there and welcome questions.